Do you hear me, basically?
Nope.
No.
It doesn't really do anything, it's just for the recording, so kind of thank
Yeah.
you. And there's a microphone there.
I think it still works.
Oh, it works.
Yeah, but only through the back.
It should work. Okay, now it works. So I just hold it like this, and yeah, I'll try. Or I'll try speaking loudly. Let's see. No? Oh, no effect. Okay, I'll just try it like that. So I'm Michael, again, I'm working in Shahar Azi's lab in Hadassah Medical Center, Hebrew University, and we're going to talk about functional and effective connectivity. So you already talked about, in the last few talks about functional MRI and GLM, and the most common way of doing fMRI research. And I'm going to talk about connectivity. We'll start with the basics of why we might be interested in connectivity. We'll talk about task coactivations, which is the most basic form of connectivity analysis. I'll talk a bit about large-scale brain networks and what we know about them, and effective connectivity. And then the second part will be about resting state, which is my field. And just a disclaimer, it's an fMRI talk, although many of these methods and the ideas can be applied also to EEG and to other neuroimaging methods. I'm investigating using fMRI, so this is what I'll talk about. So, as you know, the functional MRI and the PET before it were basically used to investigate which part of the brain does what, right? Which is basic localization and mapping studies. So we got modules for many cognitive functions, but then we have a very big part of the picture which is missing, which is the connectivity, how these regions interact, whether they form large-scale systems, and the classic approach of looking at each region and delving deeper and deeper into it is, again, missing a lot of what's happening in the brain, the connectivity. So that's what we're going to talk about, and why should this be interesting to us? First, well, why are we interested in the brain? First, we want to understand the brain itself because it's interesting, just scientific curiosity, and this gives us a missing part of the picture and allows us to give these localizations context, as we will see later. Second, we want to understand ourselves. Most of us came to study neuroscience or related fields because we're interested in psychology, basically, and this gives us further measures the connectivity between regions. Allows us to investigate. We can correlate it to behavior. We can see what happens if the connectivity is related to different kinds of mental activities. And, of course, curing disease states, psychiatry, neurology, and connectivity does, a lot of the recent research is about these fields, including our own in the lab. Just to take, if we're talking about connectivity, there are two main types of connectivity. So these are two terms we will be using now: functional connectivity and effective connectivity. If we're talking about functional connectivity, we're talking about correlations between brain regions. So if we say two regions are functionally connected, what we mean is their activity is correlated across time, usually. And this is, we'll talk about the issues with interpreting this. And so basically we can look at regions, say, okay, they're related to each other in some way. This is functionally connected. Effective connectivity is actually finding what is the way by which they are connected, finding the direction of the connectivity, which region drives which other region. And obviously this is what interests us. This is what we want. Effective connectivity is always better than functional connectivity. As we will see, it's much more tricky to find, at least using functional MRI. So we'll start with the coactivation during tasks. And this is the most basic form of connectivity analysis. So let's assume we did an fMRI experiment to investigate some mental function. We did a general linear model. We have a contrast. And we see that the brain responds. Some regions of the brain respond, and this is just a random connection of papers, of activation maps. So several regions of the brain respond. For example, the precuneus, posterior cingulate, and the medial prefrontal cortex are more active when remembering autobiographical information than when doing some control condition. Okay, so this is what you talked about in the last, I guess, the last few talks. And one thing, and so this basically gives us a measure. Okay, so the posterior cingulate cortex and the medial prefrontal cortex are a part of the same system. Same brain system is involved in remembering some way. There are issues with that. We'll talk about it, but that's the basic idea. And two things you can see here immediately in this slide. One, symmetry. Okay, if you look at co-activations during tasks, usually when a region in one part of the brain, in one hemisphere, is active, there is a good chance that you will see the same region in the other hemisphere active as well. There is a very high correlation between, through the corpus callosum connections, there is a very high connectivity and very high correlation in activity during tasks between the two sides of the brain. Not always, but it's usually the case. That is a very dramatic way pattern which we see almost always. And another thing, so what's strange in this figure? Have a good look.
Which one?
All of them together.
No.
Can you see something unexpected?
Same regions are activated in medial.
So if you look here on this mental rotation task, for example, and you look here at judging if objects are graspable task, they involve almost, the activations look almost the same. And also if you look at the remembering and the reflection on self, also you see somewhat similar regions, not exactly, but this is something interesting, and we'll keep this as a teaser for later for the large-scale brain networks.
It could also depend on the way we define the task. Maybe we're
Yeah.
defining tasks as
Sure, that's true. It may be that this task of reflection on self involves remembering, and that's why we see it. But I will tell you that it happens that, I mean, we do see this, at least this very common systems. Give you a spoiler, this is the dorsal attention system involved in general attention to tasks, and this is the default mode system involved in internal mentation, in thinking about the self and things related to the self. These two systems are active across many tasks, and this is also a surprising finding, which you cannot see if you're not looking at if you're only looking at asking whether the, I don't know, region empty of the visual stream is active in this task. You will miss this whole pattern of activity. Okay, so this is the functional co-activation. So how can we measure it quantitatively, except for looking at maps and saying, okay, these regions are connected? So one very basic way is measuring the correlation of the signals across time during the task. So we can take signal, we can measure the signals from different parts of the brain, and we get, for example, from each voxel we can get the signal across time during the task, and we will see it goes up and down according to the task or not. It depends on whether this region is involved. And we can ask, okay, which of these regions are related? So how do we do it? We just compute the correlation. Correlation, Pearson correlation, it's a number from -1 to 1, and 1 is completely related. Whenever one region goes up, the other goes up. 0, no relation. -1, anti-correlation. Whenever one region goes up, the other goes down always goes down, and of course it can be anything in the middle. So we can do this, or we can do what's called seed-based connectivity. We can take one voxel or one brain region. For example, we can ask which regions are related to the hippocampus during navigation tasks. So we can take the hippocampus and do correlation to all, take the signal, the average signal of the hippocampus, do a correlation to all the voxels of the brain, and for each one measure the amount of relation of this region to the hippocampus. So this is a seed-based connectivity. The region we are using is called the seed. Okay, so this is one issue. So assume we did this. We found that the hippocampus and the prefrontal cortex, left medial prefrontal cortex, are both activated during navigation. Now, yeah. Hmm? So, okay, the lag is an important issue, right? You're saying, right, if I understood you correctly, you're asking about the lag, that hemodynamic lag can be different in different regions. It depends on the task. I mean, if it's we can consider it if we are doing a block design, for example, showing some stimulus for a long period of time, then it doesn't really matter, because anyway, this response is so slow. You will see something goes up and down, and maybe it will be half a second before or later. The response in two different regions, you will still see a very strong correlation. Usually that's the case. The correlation will be there even if you don't consider the lag. There are ways of adding a parameter to the general linear model of the lag itself, and trying to find whether these regions are connected. I mean, doing cross-correlation instead of correlation, moving the time series a bit and seeing if things are still correlated. It's also possible. In fMRI, it usually doesn't. From my experience, it usually doesn't really matter. At least unless you're doing a very, very short stimulus at each time.
I guess also that the hemodynamic response is so sluggish.
Yeah.
So short time differences would be kind of blurred the same, about the same kind of thing.
Exactly. The hemodynamic response is sort of a blurring filter. Even if there are some differences, because it just blurs them over a very long period of time. So again, in fMRI, it doesn't really matter. If you do these kinds of analysis in EEG, for example, it will matter a lot, or in intracranial. Okay, so let's get back to the example. We saw the hippocampus and the prefrontal cortex active during navigation. Now, does it mean that they are really interacting with each other? What can be another explanation? Okay, third area interacting with both of them, that's true, because again, we're talking about correlation, so it may be that a third area is driving both of them. That's true. And another thing is maybe they're dealing with different aspects of the task. Maybe they're not really part of the same system. Maybe they're, I don't know, the hippocampus is related to mental maps, to thinking about mental maps, and the prefrontal cortex is related to, I don't know, a decision of which direction to turn right now. So we see them both active during the task blocks, but we don't know if they are really related in their activity. And this is this is one caveat of using this approach. And one analysis which is trying to give a solution to this is psychophysiological interactions, PPI, in which we actually ask whether the two regions are more connected during the task than during rest. So we are not looking, I mean, if we look at the two time series of the hippocampus and the prefrontal cortex, we will see that both of them go up during the task and both of them go down when not during the task. But again, maybe during this block, they are not really correlated. So we can there are several ways of doing this, but one way is taking the. I'll not go too much into the mathematics and the details, but basically we are taking the regressor related to the task and the time series of this region, and we are multiplying one by the other, and then we will get, because the task, if we are taking care that the task is between, the mean, that the mean is 0, then the new regressor, the red one, will be anti-correlated to the time series when not inside the task block, and correlated during the task block. And then if we take another region and we ask whether it's correlated, then if it's correlated to this region all throughout the task and throughout the resting state, we will not see anything. If it's correlated more during these parts, we will see a stronger relation.
Are you talking about the interaction factor?
Again?
The algorithm that you're adding is especially interaction?
Yeah, so it's an interaction term, and we are actually, we're looking at the interaction between the task and the activity in this, and the connectivity between these two regions. We're actually trying to put this connectivity in context. We're asking whether this connectivity is stronger in one context, in one condition, than the other. Again, if you want, we can delve deeper into this, but I just want you to get a basic idea of what we do in this analysis. And this enables us to get a bit more knowledge about whether these two regions are really interacting during the task, are really correlated also during the task and not related to the task independently. Okay, the limitation. So you already said it. What is the biggest problem with analysis based on correlation, either the PPI or the functional connectivity? We don't know if there isn't a third factor driving this correlation. So, if we see, for example, two regions correlated in their activity, in the ventral and dorsal visual streams, they may not directly speak to each other. And there may be a third region driving the activity in both of them. And this is a very strong issue. We know there is a relation. They are part of the same system, but we don't know that they could immediately interact. This is in contrast to structural connectivity. If we see, white matter fiber going straight through one region A to region B, we know they are connected. Here we don't. We know they are. So whenever someone says regions are functionally connected, just keep in mind, it doesn't mean they really have to be connected. They have to be structurally connected. They don't have to be exactly connected in the strong sense. They're just related.
In the strike, connected and right, cultivated.
Yeah. Yeah, a lot of people say that, this field of functional connectivity should be called functional correlation, just to be more precise and more careful, because it's very easy to, it's very easy to confuse the things. Say, okay, we see these two reasons, and we'll talk about it. There are even more fine details with this interpretation of connectivity. It's a big issue. But again, if we do see that there is a relation between these two regions, the relation is there.
Would you raise the issue of, so this third thing that could drive them would be also physiological, that they have the same brain, because let's speak about that.
Okay, I have it at the end of
Let's keep it in.
but yeah, basically just in one word, there is a more discussion about it when we'll talk about resting state correlations and about the caveats. But basically, what Aviv is saying is a very big issue in the field. If, for example, the subject is moving, then two regions of the brain may have spiking in the activity or spiking in the signal at the same time, and this will raise the correlation. We will think that these regions are correlated. Although if anything that changes the signal across the brain at the same time It can be breathing, it can be movement, it can be scanner noise. The scanner has a glitch or something. We will see these regions as more correlated. And this is also a big issue, and we'll talk about it later. Okay, so just to summarize this part of coactivations, we were talking about regions coactive during task. We talked a bit about this enabling us to look at this at the system level, at looking not only at activations, but a part of which system do they belong to, and we'll talk about it a bit more in a few slides. The two methods I showed you are functional connectivity, correlation-based functional connectivity, or seed-based functional connectivity, where we take one region, we ask for the rest of the brain which parts are correlated to it. And we talked about PPI, which is a bit checking whether this correlation depends on context or changes with the context of the task. And the limitation, again, we don't have a direct interpretation of these regions are connected, and we don't have causality, of course. We don't know which region drives the other, even if they are connected. Questions so far? Okay. So let's talk about the effective connectivity. Like we said, this is the holy grail. We want to be able to know which region is activating which other region. And we want to know the causation, we want to know the direction, we want the full picture. And there are two basic approaches of tackling this thing. I'll give two examples. One is autoregressive models, a more simple mathematical approach, which is simplified by Granger causality. And the other is a model-driven approach, like dynamic causal modeling. So those are two ways of trying to understand which regions drive which other in the activity. Both of them have problems as well, but we'll talk about it. So, okay. Granger causality. Granger was an economist, actually, invented, I think he won the Nobel Prize for this analysis of causality. He was interested in various factors in the market and whether they influence one another, but this can be applied also to many other fields. And the basic idea is, okay, let's assume we have signals from two regions. The basic question is whether we can predict the activity in one region based on the other region, at previous time points. Okay? So whether if we have a region X and region Y here, so we can see that X precedes Y in its activity. And this means basically if X precedes Y in its activity, we can say, okay, then X drives Y. There is delay. So that's a very basic and simple approach. The way of doing it is actually doing sort of cross-correlation, like moving the time series of X across several time points, putting it all in a regression model, and asking whether there is evidence that if we move X, for example, by 2 time points, or by 1 time point, or by 3, whether it can help us to predict the activity in Y above chance. So that's a very basic, the basic approach. It's useful. It is a bit limited because we only look at two regions and we only look at one type of interaction, which is correlation across time. So this is the Granger causality. Dynamic causal modeling is an example of another approach developed by Carl Freestone in UCL, the guru of fMRI, I think. Yeah, the big guru. Carl Freestone is sort of introduced general linear models into fMRI analysis. He built the SPM software. I think he's the most cited researcher in neuroscience.
The Granger causality doesn't even solve the basic, logically it's a bit flawed, isn't it?
doesn't rule out the fact that there might be a third driving region. Maybe
Yeah.
just one takes
True.
longer to get there.
True. That's another issue. I didn't talk about it, but it's really true. If we look at two regions and we see that one precedes the other in its activity, it may be that the information flows through a third region. We do know.
Not even through a third region. It could be a first location that drives both, but it takes longer.
True. Yeah, it gives us a very partial picture.
Yeah.
And the DCM is trying to answer this, trying to give a partial answer to this. And the basic idea is, okay, let's take a group of regions. I don't know, let's take 5 regions we are interested in, or 3 regions in this example, and try to guess all the possible combinations of activations from one region to another. Okay, let's say region 1 drives region 2 and 3, or region 1 drives region 2 and 2 drives region 3, etc. All the possible models. And let's put it into a very complex model with a lot of parameters of how, if this is so, how should the activity look like. And they have, in the DCM, they have very complex models. They actually go back to the neuronal level and the layers in the cortex. Say, okay, so the information has to go from this layer to this layer, and but this layer has a modulating effect. It's a very complex model with a lot of parameters, and they're trying to get for each of these possible models how the activity would look like. If we put an activity, if one region is active, what will be the activity in the others? And then the basic idea is checking which model is the most plausible. We take each model and we do a Bayesian approach, asking which model is the most fitting to our data. So if we find one of them, we say this is the model with the evidence points to this model being more plausible than the others. What's the problem with this issue? Again, even if we take many regions, we may not guess everything, and this may have big effects on the model. I mean, there may be other regions interacting with these regions. There may be other types of interactions in which we don't really observe, and it's still problematic. In DCM, in its current implementation, in SPM, we can enter up to 8 cortical regions, and this is, again, a bit limited if we look at the brain and what's happening there. And I think this is the reason that these methods didn't really get a lot of, not a lot of groups are using these methods of causality. This is one reason. And the second thing with causality is that there are very basic problems with this approach. First, like you said before, the hemodynamic response may differ a bit in the lag between two regions. And in the case of causality, this has a lot of influence on our interpretation. Even, I mean, everything could be reversed if we look at one region acting before the other, but the hemodynamic response is faster in this region. Maybe the other region is still driving the first one. So this is one thing. And the more basic problem with causality, I'm influenced by Tal Golan here, that the fMRI timescale, I mean, we are measuring activity every 2 seconds usually, 2.5 seconds, half a second if we have a very multi-band fast MRI. And a lot can happen in this time frame. The regions may move information from one to the other many times over, and the whole system can do a very complex thing. So it's very tricky to try to figure out causality based on fMRI. I mean, it's very unclear that it's even possible. Connectivity, yes. Functional correlation, if two regions are more active during 5 seconds, then we will be able to see it. But what did these regions, how did they interact during this time frame? It's very hard to figure out. So this is why I'm personally cautious about these kinds of interpretation. And that's just the thing about effective connectivity. So just summarize this part. We talked about causality. We're trying to find causality. We have the grandeur causality, the mathematical based methods, or the more complex models like DCM. There are others. There are structural equation modeling, other types of approaches to this thing, and it's pretty controversial, and this field is not very large in the amount of research done here. Okay.
Do you have any sense of stability of those? Do the same thing twice? Do they get this level of?
I actually don't know. I didn't play with them a lot. Okay. So let's take.
I would say something that the scary things about those things is that in most cases when I see graduate causality used or DCM, the group who use that, they typically don't even understand the first step of what's happening in the analysis. If there are groups who are using this, it's just a pure black box.
Yeah.
There are so many assumptions.
Yeah, the DCM especially is has tons of assumptions. They have very complex models, and I don't know anyone who really knows deeply understands the assumptions underlying these models. So you either take it as a software package and put the inputs and hope it will work, or yeah, it's tricky. Okay. So let's talk about large-scale brain networks a bit. So this is a bit less methodical, but just I think it's important if we're talking about connectivity. These are some of the main paradigm shifts this approach has given us, and also the resting state, which we will talk about later. Do you take a break between? Yeah. Yeah. So 10 minutes for this, and then in the second part we'll talk about the resting state. Okay. So large-scale brain networks. So like we said before, we commonly see the same regions being activated for different tasks, which is or the same patterns across the brain, not only the same regions, which is something strange, right? It's not really expected, and this is something people began to notice more and more, that in the literature, this pattern specifically, you see it in a lot of different contrasts, a lot of different tasks, this pattern too, and others. And this sort of suggests that we have some kind of, look here on the right. This is from a review by Buckner and Carroll. Remembering, imagining the future, prospection, and the theory of mind entering the minds of other people, like trying to think what another people would do. They all activate the same region and reflection on self above, the same system. So a lot of different tasks activate sort of the same brain systems, and this leads us to think of, okay, maybe the brain is organized in this way. Maybe there are large-scale systems which have different subcomponents, but maybe we can understand them more. And this is, I think, for me, this is one of the most influential papers. I know the Yeo and Buckner paper from 2011, dividing the brain into 7 large-scale networks. Again, it's based on resting state connectivity and not task connectivity, but the findings apply. All of these appear in task-related co-activations. And basically, if we look at it, we can actually see that the whole brain, here they show 6, there is a 7th one here, 6 large-scale systems across the brain, somatomotor, those are attention, visual, salience, default, and control networks, and they actually cover the whole brain. So for each activation we see, we can say, okay, this activation is not only in the angular gyrus, it's in the part of the default mode network of the angular gyrus, for example. Yeah, you can overlap. You can download this as masks, and you can actually see for each of your activations, or you can find them in individual subject networks using resting state. And I think this is a paradigm shift in neuroscience, thinking about the large-scale systems, not only the just the activation and localization. So let's talk briefly about the systems. So basically, we have the three sort of basic sensory networks. Somatomotor, you see, if you see a region in the motor cortex active, you will very frequently see a lot of the other parts of the motor and the somatosensory cortex being active together, and also on the contralateral hemisphere. So this is somatomotor, the visual, also the visual systems, the auditory, you can say, also being active, co-active during tasks. So those are the three more basic sensory networks.
The black circle is the seed. The black circle is the seed.
Here, yes.
Function connectivity.
Yeah, this is function connectivity. We didn't talk about it yet. But
the seed in a different part of the network, it
will
have
still
slightly different.
Slightly different, but mostly the same thing. Yeah. It will be slightly different, but it will be very, very similar. If you move it around, you can play with it, and it will be quite similar.
Basically, language is not part of like a human network.
What?
Language.
So there is a network which some people call the language network, which goes around. It's tricky. The names of these systems are a bit, and of course you can. There is an issue of. So if we look at this separation into systems, we can do a more fine-grained separation. We can always go deeper into more subsystems. So for example, a part of either salience or the default mode. I think a part of the default mode, this part is usually called the language network, the lateral temporal.
Are these only cortical networks? You don't take into account others?
So yeah, so this paper actually did measure only the cortex. If you look subcortically, so the hippocampus goes with the white network you see here above. You see here behind this, so there's some kind of medial temporal network of hippocampus and its surrounding regions. There is most of the basal ganglia regions work together. So there is a basal ganglia network, sort of. In the thalamus, it's more complex because it's divided into many nuclei. So each nucleus of the thalamus can be related to different cortical networks. It's also a nice finding. So this does extend subcortically, and well, we published just published a paper about white matter being also active during this. Maybe I'll mention it in the end.
For me, it's really hard to assess how good is this. Like we can just go home, like everything is solved. I just don't understand how strong it is and.
Okay, so I don't think I have it here, but in this paper, basically, again, they did it using resting state correlation, but they saw that it took 1,000 subjects. They showed that you get the same networks if you take 500 and 500 separately. Exactly the same units. So this is very robust, and I can tell you that when I do it, it's very robust also on the individual subject level. If you put a seed for most of the networks, if you put a seed here in this part of the angular gyrus, you will almost always get this part correlated to it in every individual subject. It's a very robust phenomenon. That's why one reason that these things became so popular, these investigations. It's very easy to do even in small groups of subjects. And as I'll show you, you can find these networks not only using tasks, but also using resting state. can let a person lie in the magnet for 10 minutes, and you will see, you will get the separation into networks. We'll talk about it in a bit.
When you say robust, what is the resolution that you like described here? The native 2 millimeter that box that it was scanned, maybe we should think about after the smoothing.
So,
centimeters, what resolution is the effective resolution?
So, okay, so these regions are quite big, right? Because, I mean, if you look at this patch here, that's usually what you see. So if you take one voxel in the middle of this, you will see all of the region around it very strongly correlated to it. So basically, you can look at you can say it's a very low resolution phenomenon. Of course, there are very fine scale individual variability. I mean, because the shape of the brain is different, the functional regions are sitting a bit differently in each brain. That's just show the Idota Bor paper about. We just talked about it. So there are differences, but the basic pattern remains across all subjects. It is tricky. It is true that if you take a seed, an anatomical seed, you need to be careful and take it from one of these very large clusters in order not to fall accidentally in another network. If you want to take the angular gyrus, take a seed here and not here, because then you will maybe get to another network. So you can use different tasks will give you different networks, right? Because if you're taking task activations, so if you're giving a visual task, you will see the visual regions. If you're giving, and you will not see the somatosensory network. Again, the resting state allows us to get all of the networks together, but we do get different networks in different tasks, like we just showed here, right? So this is the dorsal attention network in these tasks, and the default mode networks in this task.
Maybe when seeing the same network in these different tasks, some of them is more clear, like visual, but for attention and control, it's more get really clear.
So yeah, I mean, again, it really depends on the tasks, and you can design. There are specific tasks which we can use as a sort of a functional localizers to identify specific networks. I mean, if you give for example, an autobiographical memory task, you will see the default mode network very strongly. It's a very good way of seeing it. If you give a task, we just talk about the dorsal attention. If you give a task of focusing attention to some region compared to not, you will see this thing very strongly. But of course, task manipulations can create differences. It's not that, like I said, it's not we solved everything. This is the only issue. The brain only does 7 things, and that's it. There are many subcompartments and different interactions between these systems. Okay, so let's just go over these networks and take a break. So if we talk about the dorsal attention network, so this is the pattern. You see it going around the parietal lobe in the superior parietal, around the inferior parietal. So it goes inside the interparietal sulcus above the superior parietal and on the 2 sides of the somatomotor network. This is the most common way of seeing it. And this is a system involved in goal-directed attention, okay? So whenever you're focusing on something, you will see these regions active. When you're focusing on something external, you have to be concentrated in a task. And so this, for example, responds really strongly to task difficulty. If you give a hard task where you have to concentrate more, you will see the system more active than a less demanding task. There is, yeah.
Is this system impaired or less active than like ADHD?
Actually, I don't know. I think Mauricio Corbetta is doing things about it, if I remember correctly, but I don't know. It's a good question.
There is tons of work about working with different groups, right? Different populations with resting state, and
talk about it in the clinical aspects of resting state. Sure. Okay, the ventral attention network, which goes sort of anterior insula, inferior frontal, inferior parietal, or supramarginal gyrus. This is a system. This is an interesting system, which is more active when you're attending to unexpected stimuli, or when you switch the attention involuntarily. This is in contrast. I mean, if we talked about the dorsal attention, which is involved in focusing your attention and being concentrated in one thing, if you hear loud noise and your attention is drawn to it, or something appears in an unexpected place, this system will be activated. So it's thought to be involved in sort of monitoring of the environment. It's a bit tricky to define the exact role. We know it's involved in unexpected stimuli. We know it's involved in assessing the salience. That's why it's also called the salience network. How robust, how interesting are stimuli. So this is another large-scale brain system. The default mode network, like we said, is, I think the best way to describe it is focusing on internal processes. Instead of focusing on the outside, focusing on internal thoughts, internal memories. So that's why it's active in theory of mind, remembering, prospection, thinking about self, other things which require you to put your concentration to the inside. Just a second, we'll just say that this system is often, we'll see it, anti-correlated to the dorsal attention system. So whenever you are focusing your attention on a task, you sort of shut down the self. You sort of remove, you're dissociating from yourself. Whenever you're thinking about the self, where you're remembering something, you are shutting out the outside. You don't have any attention to the outside, and we actually see it in the activity of these two networks, which are strongly anti-correlated. Yeah.
So how do you know that the person is actually doing these things that you ask them?
Yeah, I mean, so there are many tasks, but basically.
But here there's no task, it's just self-reflection or self.
No, so there are tasks of self-reflection. So for example, theory of mind, you can thinking about what another person would do, so you can give like a task of giving a short story, and then you have to answer as what the person will do, and you know something he doesn't. So if you ask what the person will do, you have to think of, okay, what he thinks. So you have to sort of enter into his mind. This is classical theory of mind task, compared to a story which you have to complete, which doesn't have it. So you can compare these two conditions, and one of them requires you entering into another person's mind, and another doesn't. Or, for example, you can give a task of, okay, I give you a word, cat. Remember something in your past related to a cat, and then answer a few questions about it. It's always the issue with experiments. If the subject is doing nothing, you have a problem, but we assume the goodwill of the subject and we test it in some way. But you can design tasks requiring internal attention. Okay, just the last, well, we are a bit late, so there's just the last network, the frontal-parietal control network. This is a bit, there is less evidence showing what exactly it does, but we do know that it seems to be involved in integrating. It's situated exactly in the middle between the regions of the default mode and the dorsal attention. Seems to be involved in integrating between these two systems. So whenever, here you can see, whenever you have, so there is a task of autobiographical planning, you see the default mode active, and visual spatial planning, you see the dorsal attention active, and vice versa. But if you have a task involving both, then you will see the frontal parietal control lighting up. So just summarize the 7 major brain networks. We're talking about visual, auditory, somatomotor, the default mode, which is involved in internal mentation, dorsal attention, external attention, ventral attention or salient attention to unexpected things or monitoring of the environment, and the frontoparietal control, which does some kind of juggling between the different systems. Okay. So we talked about the co-activation analysis and effective connectivity, functional connectivity. Now we get to resting state functional connectivity, which is a bit more surprising field and less well understood. But this is one of the, I don't know, in functional MRI, this field is growing exponentially every year. You see tons of resting state works being done. It's very simple to do works in this field. They give a lot of information, and it lends. There are many new surprises every year. Okay, so this is the paper that started it all. A paper by Barat Biswal in 1995. Basically, what he did was he said, "Okay, I will ask people to tap their finger." I think it was only in one hand, and he looked at the activity. So here you see the activity in the hand region of the motor cortex. He showed bilateral activity, like we said. When one region is active, the contralateral region will often be active as well. So this is nothing surprising. It's actually nice. The design was he used only one slice, so he only got this slice, and therefore he got a very PR of 250 milliseconds, which is quite nice, but just on a side note. But then it said, okay, I have another scan in which the subjects do nothing. They just lie there. They don't move their fingers. I'm not asking them to do anything. And I will say, take this region as a seed, the region I identified here, and check whether other regions are correlated to it in their activity. And we saw that indeed this region, which was co-activated with it during the task, was also correlated in its activity during rest in a very strong manner. So you saw the two regions rising and falling together, the activity, when the subject is not moving its fingers at all, which is quite surprising. Another surprising thing is that this activity in these two regions, this correlated activity, is very, very slow. It's below 0.1 hertz. If you look at this Fourier transform, it's super slow. And he could measure much higher frequencies, and he saw almost no activity in these two regions. But
I don't understand which of the areas that are being correlated, and
so
what's the meaning of the.
Okay, so you see here
the meaning of the frequency of the correlation.
So it's not the frequency of the correlation; it's the frequency of the activity during the resting state. So these regions are doing something during the resting state. The activity rises and falls. It rises and falls together in these two regions, and the activity is very, very slow. Nothing like the task-related activations we see.
And what's the reason?
So here it's two sides of the motor cortex. You see, so this here is the central sulcus. What?
A and B,
Yeah, A and B, and also C. So also this region of the middle of the homunculus, the leg region, also being active with the two of them. It's actually the whole sensory motor system is correlated during resting state. And this paper, interestingly, was not well known. If you look at the citations across time, so right now it has 5,000 citations, and it's one of the most commonly cited papers in functional connectivity. But for about 10 years, nobody noticed it until it was rediscovered after other people discovered the same phenomenon again using different methods. So this is something quite surprising, that there is correlation during the resting state. Basically, to measure this correlation, we can use the same methods we used as when we talked about tasks. So we can do this functional connectivity, check the correlation, the signal from each brain region, measure the correlation, and see which regions are correlated. We can do a seed-based approach, as before, and other approaches. And here, this is data from, I think it's Roy's brain, actually. So if you take the activity in the, so this is a resting state. This is something I just got in MATLAB. This is the motor cortex, yeah, the right and the left motor cortex of the same subject. And you see the activity is very, very highly correlated. You see the peaks and the falls going exactly together during the resting state. Also, if you look at these two regions, which are not, which are part of the frontoparietal control system, again, they are not they are not adjacent. They are separated, but their activity is very, very strongly correlated.
Just understand here, you put a seed somewhere, let's say in the red region.
So here I don't put.
You found it in the
No, so here I didn't use the seed. Here I took all of this region, averaged the activity for the voxels, all of this region, averaged the activity of all the voxels, and I'm showing both of them. So this is not a seed-based correlation. This is just measuring the correlation between two regions. Okay?
And if you did, so you assume, when you knew that these were two motor regions, if you took
Yeah.
the red region, the right motor region, you just looked at the
At a seed-based correlation, if I took the right.
Let's just look at another region which we don't think there should be a reason for a correlation.
So it depends. If it's part of the same network, you will see a very strong correlation. If it's not, you will not.
You don't see correlation at all?
It depends. Some of the networks are slightly correlated to each other, but very not like 0.7. It's very, the correlations inside each network within the different regions is super strong. So this is again something very surprising. I mean, why should it be like? Why would these regions even be active during the resting state when the subject is doing nothing related to it? And this actually, this is a figure from a famous paper by Zhang and Reichel. And I mean, if you use a seed in different parts of the brain, you actually see the dorsal attention network, the auditory, the sensory motor, the visual. So you can see all of these networks we talked about also using the correlated activity during the resting state. And this is first surprising. Second, it's very useful because if we have a way of identifying these networks in individual subjects and these networks of regions which are coactive during task, only through the resting state, we can do a short, instead of putting the subject for 1 hour in the magnet and giving him different tasks to identify the location of the different networks, we can take 10 minutes, 5 minutes even, and we usually see these things very robustly if we know where to put the seed.
So do you instruct the subject to do the same of everything?
So there are different styles. I usually instruct them to think about whatever they want and leave their eyes open. There are differences in the amount of connectivity in each network if you ask them to think about whatever they want, or if you ask them to think about something specific or not think about anything, or if the eyes are open or closed. So it does matter, but you still see the same networks.
Going with these same thoughts, and because those areas are very, I mean, the same thought of like you can replace hours of fMRI with 10 minutes resting state, but because this area is also co-localized to anatomy, you could just take the anatomy, not even do the resting state, get ready for it.
Okay, so you could use
when is this stuff, or where it's ready?
So the tricky thing about anatomy is first that we don't have any way of actually measure cytoarchitectonic divisions.
You would use, I mean, this is like very structure segmentation would work here. Find like big regions. I'm seeing like big regions that go correlate, right, in time, and that replicate very long strength in fMRI. But because they seem also to be in a very specific location, I would say maybe I could just segment the cortex, call this area, I don't know, occipital cortex, and It probably would also predict the resting state that will also predict the fMRI. That's it.
Yeah,
I don't need even the rest. I can do one step or not. I'm just asking.
So it is a common. We can do it. It is a common approach. It works for many regions. I mean, if you take, for example, anatomically the posterior cingulate cortex, you know it will be a part of the default mode network, and it will be correlated to regions in the medial prefrontal. It works less well for regions where you have anatomical variability, or for example for these regions where many networks are close together. And there the inter-individual difference in the location of functional clusters will usually be a problem. It will be harder just to decide based only on anatomy.
But those differences are reliable within something. These findings,
Yeah.
like this border, are reliable.
Yeah, if you find the networks,
Right?
exactly. If you find the DNA sites, these findings are reliable. First, you see the same networks across individuals, but slightly different in shape, but still the same general pattern. And within the same subject, you will see exactly the same if you do several different resting state scans or tasks.
My question is related to what she asked, but I mean, is rest well defined? I mean, is it like everything you do that is not a task is rest? And then how can you interpret the results that?
A good issue. We'll talk about it in a second. What's actually happening during rest? Is it actually rest? Is it not? So this is one way of doing this. Before we talk about the meaning, significance of these correlations and why are they interesting and what can we do with this, we'll just talk about another way of identifying the connectivity during resting state between regions. And I mean, one problem of the seed-based approach is that we need to know where to put the seed, and we need to we need to have a sort of hypothesis. I mean, we can do like put seeds in all voxels and check the connectivity, but then it's a bit complex to combine all the information. And one way of doing a combination in the same one analysis, which gives us all the networks without putting any seed, is ICA, independent component analysis. Basically, the idea is in ICA is it's a mathematical. Assume you have a cocktail party, you have many people speaking together, and you have several microphones located in different parts of the room, recording the conversations from different locations. The idea of ICA is enabling to disentangle the different talks. So each of these microphones will hear. So I assume there are 10 conversations going on all together. So each microphone records all of them. But we assume that one microphone will record mostly. One talk it will hear very strongly, another talk it will not hear at all, things like that. And if we combine the information from all of them, again, we'll not go into the mathematics, but the basic idea is disentangling the independent components that occur in this thing, and how does it apply to functional MRI? So basically, we can ask, okay, let's take. So each voxel is sort of a microphone, and each network has its own conversation, right? Because all the parts of the network have correlated activity. So you can say, okay, we have 7 or 10, I don't know how many networks, how many conversations going on, and let's try to use this algorithm to understand which conversations are going on, and to dissociate them, and then to find which voxels are related to each one are most strongly related to each one of those conversations. And so this is the basic, just hand-waving explanation of ICA. And again, the advantage is that it's data-driven. We get the independent components. We get all of them together. The disadvantage is that some conversations that are going on in the brain or some things, some of the signals which we see across many places are related to noise. For example, we see some components which are related to some subject motion, to other types of noise, to the cerebrospinal fluid, things like that. We see many. We usually, if we let the ICA take out like 30 components, we'll see maybe 5 networks and 25 noise components, and we have to manually check which one is actually a network and which one is not. And usually visual inspection. You just
you recognize it already.
Yeah, you look, you say, okay, this looks like the default mode network, and it's okay. There are ways of matching to common templates, of course, but
you assume that you have these networks when you start work. I mean, when you do the estimation.
yeah.
But not in your ICA. ICA doesn't know.
The ICA doesn't. I mean, the ICA just tries to disentangle, to find things in the data. Sometimes it even finds, you'll give 30 and it will find 2 networks and 28 components of noise. So it would be, and you need to give many more to find these networks. It's a bit tricky to play with this.
It's like PCA, in the sense that those are the ones that explain most of the variants actually explain by noise.
So it is related to PCA. I don't remember the exact
the
idea, but the idea is to
go with how much they explain of the variants, right? So the ordering or the ranking.
Yeah, so
explain most of the variants and so on.
Yeah, but then maybe some of the noise components explain more of the variants across the whole brain or the whole image. Then it's true. Usually you get some of the networks as the first components, but some of the networks are very low down in their ranking.
Can you explain what would you count as noise? Like, is anything that's not the 7 main networks is noise?
So usually, I mean, if you're using the method of visual inspection, usually you're just looking. Mostly the noise components have a lot of activity outside the brain, for example, or in the CSF, and then it's not neuronal activity. Or you see some things which look like across a whole slice or something. I mean, usually when you see a component which includes a lot of gray matter and defined regions, clusters inside it, you treat it as a real functional component. Again, this element of individual identification is very problematic.
When you run these experiments, do you find other networks which are not studied?
So like we said, these 7 networks you can find subnetworks. So sometimes, for example, the default mode network, you will see these 2 parts dissociated. The medial prefrontal cortex on both sides of the brain as one network and the posterior cingulate as another network. It really depends on the strength and the specific activity. That's one of the main problem in my experience with this method is that it requires a lot of manual tweaking to get exactly the thing you want, and then you're a bit biased because you're doing the manual tweaking. I mean, you're looking for what you want to find. So if it's something, I mean, the default mode network is usually the strongest. You usually can find it and you say, okay, this is it, but then you don't know if you would change the number of components to 50 instead of 30, maybe you'll find the default mode network, but only a sub-part of it. So it's a bit tricky using it. So there is no getting away from, I mean, there is no really data-driven approach to get these networks. Either you define seeds in advance or you use the ICA and you manually inspect.
So what are the inputs to the ICA?
Just the time courses of all the voxels in the image.
So
and then
as many components as the voxel?
No, you decide on how many components you want, exactly as in PCI. I mean, you can give as many input vectors as you like, but then you have to decide on a small number of components. Otherwise, it will also be computationally impossible.
If each voxel is a conversation, then how do you trace it back to the start?
So basically, what the ICA does is trying to identify the conversations, the common conversations across the brain. So it looks at voxels as recorders, and it says one advantage is, I didn't speak about it, is that the same voxel can contain two different conversations. I mean, maybe sometimes some of the networks you see a bit of overlap between them in different networks. It's a tricky field to investigate, and I don't want to go really deeply into it, but basically what you give is the voxels. You assume many voxels are recording the same conversation or several conversations together, and you're trying to find which are the common conversations, sort of like a clustering algorithm. And then it gives independent component analysis instead of principal component analysis, because it tries to find components that are as independent of each other as possible. Okay?
So does it take into account that if one area talks to another, then it will be activated earlier? They're not activated at the same time,
so this assumes that they are activated at the same time. It doesn't. This is not effective connectivity. It's functional. So we assume they are active at the same time, or mostly at the same time. Like we said, it's a reasonable assumption because of the time scale of fMRI and the smoothing by the hemodynamic response function.
Is any smoothing pre-processing, or do you use any spatial regularization for the type of component that you will get?
So you can smoothing during the preprocessing is your choice. During this analysis, we didn't talk about the preprocessing at all, but basically it's the same as the other fMRI. As the other things you do in fMRI, you do some kind of motion correction, you do normalization to a template usually, unless you want to look at if you want to compare the subjects, you have to do a normalization. And usually you do smoothing. It improves the signal, because smoothing does improve the signal. You see the same effects if you don't do smoothing. But
the ICA itself has
more noise.
Like a special.
The
ICA. We look for only for there's another rule essentially that say can look only for things which are not sort of pepper or something like that, or just happens to be that they also end up being continuous and like it's very
no, it doesn't rely on the component thing on being continuous. Many of the components of the noise components you see are just isolated boxes all over. So it doesn't. It also finds things which are not spatially continuous.
There's no spatial information provided to the ICA.
Yeah.
That was
semantic.
Mm-hmm.
But you can. Okay, you cannot.
Yeah, you can shuffle all the time courses and still find the components. I guess I never tried it. Okay, so this was ICA. This is just what I told you about. Again, the anti-correlation, if you look at the regions related to the dorsal attention and the default mode network, very adjacent but anti-correlated in their activity. This is from a famous paper by Fox and I think it was Buckner group. Yeah. And again, you can see the regions, anti-correlations between the default mode network and the dorsal attention network. Like we said, these networks are anti-correlated during tasks, because when you're applying external attention and internal attention, it's a competition in resources, sort of. Just to show you, this is a correlation between structural connections and functional connections. I could have shown it earlier. There is a correlation. So regions, when we are talking about functional connectivity, resting state functional connectivity, the regions which we see as more connected are usually structurally connected. But correlation is not, there are many outliers. So this is something important to keep in mind. Structural connectivity and functional connectivity are not the same, but they are related. Just mention, we don't have much time, but I just mentioned that this slow activity in these networks also exists during tasks. So we found it on resting state, but if you look at task states at the rest periods of the task, if you have a block and you have rest period in the middle, you will still be able to identify these networks. And also if you look at the task time courses and just regress out the task itself, the effect of the task, you will still see these networks, this very slow activity in the background. So this task and the resting state are related, and just say that it's, and then I'll answer you, the task and the rest are they affect each other. I mean, for task performance affect the resting state. If you scan a subject, you do a resting state scan, you give him a task involving strongly brain region, one brain system, and then you do another resting state task. During the following resting state task, these regions will be more connected. So the rest is not exactly static in the strength of connectivity, but it is affected by tasks. A lot of Rafi's work, in Weizmann, Rafi Malach. Okay, so maybe I'll ask you, what can this activity mean in resting state? Before I show you the theory. Why should there be activity during the resting state? Or maybe you had a question? Okay. Any ideas? Why should these regions be related to each other during resting state?
Yes, we have typical mental activity.
Okay, mental activity is one explanation. It's a good one. So you're saying, okay, maybe now you're lying with your eyes open, you're not doing something. Maybe some of the time you're thinking about yourself, some of the time you're paying attention to the scanner, some of the time you're thinking about moving or moving your body a bit. Then we will see these things because of the rises and falls related to mental activity. So this is one of the common explanations. One issue with this is that we see it also under anesthesia. We see these correlations. But still, it doesn't mean that it's not part of what we usually see in resting state.
My idea kind of the anesthesia part doesn't add up to this, but maybe working memory if the task affects it.
Yeah, okay, so if we had a task, or even if we don't, working memory, exactly, it's the same, sort of the same thing. Maybe at some time where the working memory system is working more strongly because we have some thoughts requiring working memory more than others, and then we'll see the rise and fall in the working memory regions together. That's true.
Spontaneous activity affected, that could be also seen, that also shows the connectivity between the areas.
Oh, so that's one of the interesting explanations. I think this is Rafi's favorite. So that's if you have regions which are connected or which are usually active during task, it may be that when one region is active, it will the other region will also be active. The threshold of one region activating the other will be low. And then if you put random noise into a system with very strong connections, when one region is active just because of random noise, the other region may be also activated. And this may be one of the things, just random noise entering the systems and activating the things in the same manner as they are functionally collected. And just want to show you some more things in the 5 minutes we have left. So the last possible explanation is the maintenance activity, some sort of metabolic or neuronal activity which is helping the brain in some way maintain its metabolism, homeostasis in these functionally related regions. But the truth is, we don't know, and this is one of the most tricky issues about resting states. We don't know what we are measuring, actually. We see the correlations, we know they're related to tasks, we know they change after task states, they change between people in relation to behavior. I'll show you quickly now, but we don't know exactly what it is.
Why then use it?
Okay, so why use it? So let's go back to the three things we said before. Why are we interested in connectivity? First, as additional things to correlate to behavior and to task performance. Second, to investigate and understand disease states. And third, to actually understand what happens in tasks. So I think this is one of the most dramatic findings in the literature. If you understand the default mode, default mode network activity goes down in Alzheimer's disease, in patients with Alzheimer's disease, or not activity, the connectivity, activity and connectivity, the connectivity between different parts of the default mode. And if you look at the regions most strongly affected by Alzheimer's disease, so this is purely amyloid deposition, this is purely anatomical data, pathophysiological data, and you look at the resting state network, at the default mode network, you see almost exactly the same regions. This is, we still don't understand why it happens, but it's super strong. And these are the regions undergoing atrophy most strongly. So this is one interesting finding. We see also that in schizophrenia, for example, there is more disconnection. Here the lines reflect the strength of connectivity between different brain regions. So there is a disconnection between regions more strongly. This is one of our works in NMDA receptor encephalitis, a disease targeting the NMDA receptors in the brain. We see frontotemporal and frontoparietal connectivity differences, and differences in the specific brain networks, and this relates to the symptoms of the subject. So subjects which have more psychotic symptoms have more disturbed frontoparietal connectivity, for example. So one reason to use it is to understand, even if we don't understand exactly what's happening there, we know the location of the connectivity disturbances of the brain system which is disturbed in specific disease states, and we may understand something which we didn't know about the disease. One additional advantage is it's very simple to let a subject just lie in the magnet and do nothing. Even vegetative patients, even patients which have big difficulty doing tasks, they can do the resting state, usually. Another thing, this is Idota Vor's paper from Science. You can predict task performance. Actually, so here you see performance of subjects in different tasks. These are different subjects. So, for example, here there is a social theory of mind task, and you see the pattern of activity in each subject, which is quite different. And this is the pattern of activity predicted by a resting state. So basically, the resting state correlations sort of contain the information about which brain regions will be active during different tasks. And you can just use this very short paradigm to understand which brain regions are collected and which involved in different tasks. And this it's not only the location, but also the strength of the connectivity can predict behavior. This is a paper by the group of Brian Levine. People who have more strong correlations between the medial temporal lobe, the hippocampus, and visual regions, when you ask them to remember things, they remember them more vividly than people with lower correlations. And people with more medial temporal lobe to prefrontal cortex connectivity, when you ask them to remember things, they remember more semantic details. So we can actually, this resting state correlation, this connectivity between regions actually tells us a lot about what the brain does, what the brain will do when performing tasks, and even about behavior. Okay, I'll skip this. Just tell you that we found also, I mean, this field is growing and offers many surprises, such as correlations inside the weight matter, resting state networks in the cerebellum. There are a lot of things happening, and a lot of interesting new findings, and it's a very popular and growing field. This is yours. Okay, just 2 minutes to talk about caveats and the problems of resting state connectivity. So we said first, we don't know what we are measuring, right? We see the correlations. They're very low. They don't seem like functional activity as we know it. We don't really know what it is. It can help us get localizations of disturbances, but not exactly what's happening there. And one problem is that these things are very strongly influenced by noise. Like we said, if the subject moves a lot of the regions in his brain will be more correlated than a subject that doesn't move. So this is first a problem. There were works, for example, showing that in young kids the brain is more connected, and with development it gets more is more globally connected, and in development it gets more separated into networks. And this probably later it was found that it's probably related to the fact that the kids moved more in the magnet. This is a Viv's paper from 2009. They took a pig's brain, shaked it inside the magnet, and showed that you can see quite nice systems correlations only through movement of a dead pig's brain. So this motion, we have a lot of, we have a lot of ways of controlling for this, but this is still a very tricky issue, the noise that enters into the system. When we have task, if we have different blocks inside the task, we can average the noise out. In resting state, we cannot. We just we see the correlations, and we have no way of knowing whether it's related to other noise signals or to our own interesting signal. And another issue is interpretation. So if you take, for example, let's say you see amygdala prefrontal connectivity in healthy subjects of 0.6, in anxiety 0.8, and in psychopaths 0.2, you can say, okay, maybe they are psychopaths because their amygdala and prefrontal cortex are disconnected and they are not connected to their emotions, or something like that. But it could be also many different things. Maybe the mental activity, maybe the anxiety subjects are thinking more, are having much more emotional thoughts, and then these regions are more activated. Maybe the amygdala is not functioning in these psychopaths. It's not that the connectivity is damaged. Maybe one of the regions is not functioning, and then the connectivity will seem different. We cannot separate the activity from the connectivity. Again, we can infer where the problem was. We cannot exactly say what happens there using functional connectivity alone. And okay, not to go into the SNR problem, but it's also tricky. So that's it. That's just resting state correlations. We talked about them. They mimic the co-activations. They have very low frequency. They're very robust across subjects. They give us a lot of interesting information about behavior, about disease states, but the interpretation of what exactly happens there is tricky, and we have to be very careful about noise sources, which may influence and may create artificial effects, which we are not interested in. But it's still a very exciting field with a lot of happening and going on in it. And that's it. Thanks.